Welcome everyone. My name is Jason Sprowl, an associate professor in the Department of Pharmaceutical Sciences at the University of Buffalo. And I'm going to talk today about the novel benefits and consequences associated with altering transporter activity during cancer treatment. So there's a few learning objectives that I'd like to go over first before we begin. The first is by the end of this, I'm hoping you'll be able to identify the risk of tyrosine kinase inhibitors involved in drug interactions with OATP1B substrates. You should be able to describe the regulation of solute carriers by tyrosine kinase activity and phosphorylation, as well as its sensitivity to tyrosine kinase inhibitors. You should also be able to interpret the benefit of using tyrosine kinase inhibitors to manipulate solute carriers involved in platinum toxicity. So first, to start, why do we want to go over altering transporters and improve cancer therapy? Well, there's going to be approximately 2 million new cancer cases that are expected to occur in the United States alone this year, and this has been reported by the NIH. And of that, approximately just over 600,000 individuals are expected to die as a result of the disease. While that may seem quite concerning, it is great to know, though, that the 5-year survival rate has generally increased to approximately about 70% of patients, which is fantastic. And this has largely been attributed, at least in part, to new drugs being made available. And this is an excellent publication that was just published last year, showing this massive increase of the number of drugs we have available and how many more have been approved. However, our increased rate of more drugs being available does bring about a concern that we're not quite mastering the drugs that we have before we kind of move on to another one. And this poor understanding of drug properties does increase various risks, two of which are, one, drug toxicity. And this is particularly a concern with our increased survival rate, because now patients are surviving, but they have to deal with the toxicities associated with treatment. You also have an increased risk of drug-drug interactions, of which cancer patients are very susceptible to. It's estimated that approximately a third of cancer patients experience some form of drug interaction, and that drug interactions may actually attribute to about 4% of deaths. So there's various factors that can contribute to drug toxicity or drug interactions, drug response. But I'm going to focus a little bit today on the changes associated with absorption, disposition, metabolism, and elimination, often abbreviated to ADME, because it's often involved in patient response and variability. But ADME also is largely often dependent on membrane transporters. And just showing here a figure of what drug transporters do is they can recognize molecules such as drugs and move them across what should be a semi or selectively permeable cell membrane. So you can imagine that in order to get a drug to its target, it'll have to move into a cell, or if it's involved in clearance, it's going to have to move through such a protein to be eventually cleared from the body. So variable transporter activity has been shown to alter ADME, and altered ADME being attributed to changes in risk of drug toxicity and efficacy. And a good example of that is shown here, a transporter known to be involved in, for example, maybe liver uptake, so that there will be eventual clearance. Once the drug moves in through the transporter to the liver, it can then be cleared into the bile. But if the transport is not working for a genetic reason, or like genetic variability, or maybe a inhibitor preventing that protein from functioning, the victim drug can't move through, and therefore just accumulates in the blood, increasing systemic exposure. And what this can then do is promote toxicity or adverse events. The problem is with all of this is that, while yes, I give you a nice little depiction of what can happen in one particular example, unfortunately, transport proteins are among the most understudied family of proteins in the human system. And this is particularly true for solute carriers, or SLCs. And in fact, how SLCs are regulated, or how many of the members can impact treatment or patient response, is very poorly understood. So I'm going to give you an example of poor understanding and what has happened in the clinic, and how we've actually worked to kind of explain this. So tyrosine kinase inhibitors, which are relatively new small molecules, they've been around since the early 2000s, and more have been approved each year. But these are chronically orally administered drugs that are used to treat cancer and various other diseases as well. But the problem is that chronic drugs, as you may expect, are often associated with polypharmacy, especially in older patients, or cancer patients, more precisely. But what was noted with tyrosine kinase inhibitors is that there were various unexpected and sometimes life-threatening drug-drug interactions that were reported, and this is despite trying to avoid metabolism-mediated drug interactions, which are quite well understood and known. So just shown here are from various different studies that were published, all cited below. And you can see various tyrosine kinase inhibitors such as pazopanib, dabrafenib, fostamatinib, regorafenib, just to name a few, being co-administered with various statin drugs as well as paclitaxel. And you can see the consequence of these combinations leading to increased rates of rhabdomyolysis, which is life-threatening, as well as tenfold increased level of simvastatin systemic concentrations, threefold, various increases in drug exposure, all of which can lead to myopathy, rhabdomyolysis in these particular cases, and life-threatening. But what was surprising out of this was that none of these drug-drug interactions were expected, hence being given to patients, but they weren't expected based on the preclinical models that were used before administering to patients. So what could be the reason for it? So we noted that a lot of these drug interactions involve statins and taxanes, which are actually substrates or recognized by the liver uptake transporter known as the organic anion transporting polypeptide, OATP1B1. So this is just best shown here in a publication in 2012 that looks at rosuvastatin uptake in cells that have OATP1B1 versus those that don't, or mock transfected cells. And you can see an increase cellular accumulation in cells that have OATP1B1 of rosuvastatin. Now, great that statins, and we've known this for a while, that they're substrates and they need this transporter to be moved into the liver and then for eventual clearance. But you might be asking, how could tyrosine kinase inhibitors impact OATP1B1 function, especially given that we know they're not great substrates themselves. Various tyrosine kinase inhibitors have been looked at, and most are not considered great substrates. So competitive inhibition, or competing for binding sites of the transporter, is not really expected, hence why they were given to patients. However, when you actually do experimental analysis, and this was done using overexpressing HEC293 cells, overexpressing OATP1B1 HEC293 cells, you can see that numerous tyrosine kinase inhibitors could diminish OATP1B1 activity. And this activity is measured using a fluorescent substrate. And among all the tyrosine kinase inhibitors, we noted that nilotinib was by far the most potent. In fact, if you do a concentration curve, you can see that the IC50, or the inhibitory concentration, of which 50% of transport function is lost, is approximately 1 micromolar, which is below the approximate 4 micromolar Cmax, or maximum exposure concentration, that occurs in patients. And moving away from just overexpressing cell lines, this is data showing uptake, or OATP1B activity, in human hepatocytes. And you can see here that nilotinib was able to reduce the uptake of an OATP1B1 substrate known as estradiol beta-glucuronide, but that was not the case with ruxolitinib, which we had previously shown did not have inhibitory properties against OATP1B1. And we were able to also confirm that a drug-drug interaction can occur between nilotinib and a statin. So this was done in vivo, where we provided nilotinib at 50 milligrams per kilogram, orally administered 30 minutes before rosuvastatin at 10 milligrams per kilogram, also orally administered. And you can see here the resulting systemic exposures. So looking at rosuvastatin concentrations in the blood in mice that have vehicle versus those that were exposed to nilotinib, and you can see that those that were exposed to nilotinib had a much greater systemic exposure after this single dose of the two drugs, therefore confirming what was seen in the clinic with certain tyrosine kinase inhibitors and statins. So great that we've now confirmed that it happens. Obviously, this is a great risk, but we've also asked why, or how can these tyrosine kinase inhibitors be contributing to this event? Where the importance of this OATP1B transporter is well known, the role of its regulation is poorly understood. And this is particularly true for PTMs, or post-translational modifications, and how they regulate the transporter. It's just really, really poorly understood. So given that we were working with tyrosine kinase inhibitors, we were very interested on the phosphorylation status, because kinases are involved in phosphorylation. So we decided to look at whether OATP1B1 was tyrosine phosphorylated. And using mass spec-based proteomic analysis, we were able to show that there are numerous phosphorylated or tyrosine phosphorylated sites located within this protein, and that there's two particular sites that were highly phosphorylated, Y640 and tyrosine 645. And we also noticed that when you exposed this transporter to nilotinib, that these phosphorylation sites actually decreased in abundance. So there was a significant decrease, particularly for Y645, in the presence of nilotinib, indicating that you're losing the phosphorylation status following treatment. And these particular sites seem to be important, and the phosphorylation status itself is particularly important, because if we mutate this site to a phenylalanine from a tyrosine, which maintains the bulky phenyl structure, but you don't have the you don't have the alcohol group associated with the benzene ring. As a result, you actually lose transport activity in either case. So this really indicates that Y640 and Y645 are very important for transport function. Now, in regards to specifics of these sites, we know it's located near transmembrane domain 12. Looking at the newly available crystal structure, this would indicate that it's really close to the internal membrane component shown here in the circle. It's a highly conserved location, and the phosphorylation status might be involved in structural shifts. So it's not close to the binding site of substrates, but that doesn't mean we can't have structural shifts that are involved in the movement during the transport process or in order to recognize. But further study is going to be needed to clarify what exactly is happening. But what we do know is that this area is accessible to kinases that would be present within the cell. As to which one, this is where we've done a little bit further analysis. In particular, we did an siRNA screen that targeted 779 different kinases, one at a time. And we looked at any that would reduce OATP1B1 activity, where you diminish the kinase expression, but without impacting the cell viability or cell survival. And from this analysis, we were able to show that an siRNA that targeted these kinases in red were actually able to reduce OATP1B1 activity. But we focused more on LIN, because among all of these kinases that I'm showing, this is a tyrosine kinase that is actually shown to be sensitive to numerous OATP1B1 inhibiting tyrosine kinase inhibitors, nilotinib being one of them. So looking at a further assessment, we actually used our overexpressing HEC293 cells, again, the model that we did our original screen, and we subjected them to siRNA that targets LIN, meaning that we were able to successfully knock down LIN expression, and you can see shown here where we lost about 50% of LIN expression. And this 50% reduction in expression was resulted in loss of approximately 50% of OATP1B1 activity. indicating that Lin is a major regulator of OATP1B1 function. So in summary, what we think is happening is that OATP1B1 is a tyrosine phosphorylated protein, and that this phosphorylation event is required for protein function. We know that Lin, a tyrosine kinase that is sensitive to numerous tyrosine kinase inhibitors, is involved in the regulation of OATP1B1 function. So obviously, tyrosine kinase exposure seems that targets Lin seems to be able to inhibit OATP1B1. And I really want to highlight that this actually is more devastating than just targeting OATP1B1. And the reason for this is that we did a follow-up study on a homolog of OATP1B1. So there's actually a partially redundant or sister protein that is 80% homologous to OATP1B1, known as OATP1B3. So this can often compensate. There's a lot of substrates that are recognized by OATP1B1 that can also be recognized by OATP1B3. So we naturally asked if this transporter is sensitive to tyrosine kinase inhibitors as well. And perhaps unsurprisingly, we confirmed that this was the case, that OATP1B3 function was sensitive to numerous tyrosine kinase inhibitors. And this right here is showing data from overexpressing HEC293 cells of this particular transporter using, again, a fluorescent substrate, 8 fluorescent cyclic AMP. And again, nilotinib was identified as the most potent inhibitor. Again, doing an IC50 curve or exposure to various concentrations, we were able to show that the concentration required to inhibit 50% function was, again, approximately 1 micromolar or just under. And again, using human hepatocytes, we were able to show that hepatocyte uptake of CCK8, which is an OATP1B3 specific substrate, was reduced in the presence of nilotinib. And this being 4 micromolar of nilotinib, which again is approximately that Cmax or maximum exposure in patients. So naturally, given that we saw that similarity with OATP1B1 and 1B3, we were curious if Lin also regulated OATP1B3. You know, given the sequence similarities, the structural similarities, substrate similarities. So first, we asked if OATP1B3 was phosphorylated. And indeed it is phosphorylated in numerous sites. You can see actually even Y640 is also highly phosphorylated, similar to OATP1B1. Y645 actually is not present in OATP1B3, so I just want to highlight that. But yeah, in regards to Lin regulating OATP1B3, we did a similar study to what we did with OATP1B1, showing that use of an siRNA molecule that targets Lin, which reduced Lin expression, was successful also in reducing OATP1B3 activity, and that's shown here. So in summary, this means that you can actually have a loss of both OATP1B transporters in the liver, which brings about a higher risk of drug-drug interactions than even originally anticipated. Through our studies. This may also show why you have high rates of hyperbilirubinemia with various tyrosine kinase inhibitors in patients, because conjugated bilirubin is a substrate of these transporters, and loss of both can actually lead to accumulation of bilirubin in the systemic circulation. We're also curious now too if variability in Lin kinase activity may contribute to interpatient variability of drug response. There's a lot of substrates that are recognized by OATP1B transporters, and so if Lin is variable from patient to patient, this may actually contribute to variability that we see with different patients. So obviously, there's going to be further investigation required. We're actually already using human hepatocyte models, humanized mouse models, and PBPK PD modeling to fully characterize the risk that may exist with patients. So while I just now went over how this kind of a drug interaction can contribute to toxicity of victim drugs, I want to explain how this knowledge can actually be a benefit to treatment options as well. And to do this, I'm going to focus on OCT2 and platinum toxicity. So OCT2 stands for the organic cation transporter 2, or OCT2, which is a protein or a transporter that is selectively expressed in few tissues, three of which are actually in the proximal tubules of the kidney, cochlea of the ear, and the dorsal root ganglia of the peripheral nervous system. And it's actually well known that two OCT2 substrates are cisplatin and oxaliplatin. So meaning that expression of this transporter results in increased accumulation of cisplatin or oxaliplatin. And it's important to note that there are toxicities associated with cisplatin and oxaliplatin that include nephrotoxicity and ototoxicity, which are prevalent in cisplatin-exposed patients, as well as neurotoxicity, which occurs in both cisplatin and oxaliplatin treated patients. But you'll note that these toxicities all link to where OCT2 is actually expressed. And as such, it's not quite surprising. I'm not going to go over this in great detail due to time, but it has been shown in the literature that diminished OCT2 function, either using genetic deficient models or mouse models, or even patient genetic variability, OCT2 seems to provide protection, if defective, from platinum toxicity. So as such, if you would think that maybe use of an OCT2 inhibitor could actually be used in combination with platinum drugs to prevent toxicity. And this is where I want to highlight that similar to what we saw with OATP1B transporters, numerous tyrosine kinase inhibitors have been identified as potent OCT2 inhibitors as well. So this is just showing a screen of various different tyrosine kinase inhibitors reducing OCT2 function as measured in overexpressing HEC293 cells with a fluorescent substrate known as ASP. And you can see here that amongst the TKIs present, that dasatinib is the most potent inhibitor, and actually very potent in that you only need approximately 20 nanomolar to hit the IC50 for OCT2 function. So what was kind of shown was following this in regards to mechanism is that similar to what we saw with OATPs, these tyrosine kinase inhibitors that inhibit OCT2 function seem to be able to reduce OCT2 tyrosine phosphorylation. And this was actually shown rather than mass spectrometry using a phosphotyrosine antibody following purification of the protein. So you can see by Western blot analysis that you have phosphorylation bands present with OCT2 exposed to DMSO or imipramine. Imipramine is a substrate of OCT2, so it doesn't impact its phosphorylation status. But numerous tyrosine kinase inhibitors result in loss of this phosphorylated band, meaning that phosphorylation was lost. And actually, even from there, it was shown that many of these OCT2 inhibiting tyrosine kinase inhibitors are YES1 inhibitors, meaning YES1 is a Src kinase. And this was shown to actually result in loss of OCT2 function, where if you use an siRNA molecule that targets or diminishes YES1 expression, you also get loss of OCT2 phosphorylation, as shown here by Western blot analysis. So given that we've now characterized, identified OCT2 inhibitors, as well as characterized the mechanism by which it's occurring, we sought to see if you could use YES1 inhibitors or Src inhibitors to ameliorate platinum toxicity. And this actually looked to be quite successful, although it's been limited so far to mouse models or rodent models. So far, dasatinib has been shown to reduce oxaliplatin neuropathy in mice, and this is shown here using a peripheral neuropathy von Frey-Hair test. So you can see that there's a greater sensitivity to touch in wild-type mice that have OCT2 versus those that do not have the transporter, genetic deficient mice. Don't seem to have a change in peripheral neuropathy. However, in the presence of dasatinib, your wild-type mice also don't experience peripheral neuropathy. Nilotinib, which again is also an OCT2 inhibitor and a YES1 inhibitor as well, has been shown to reduce kidney damage that happens following cisplatin exposure. And this is shown using BUN, or blood urea nitrogen levels, which is a common marker of nephrotoxicity. So after 24 hours with cisplatin treated mice, you can see increased BUN concentrations, but in the presence of nilotinib, this elevated BUN is lost, indicating protection. And then finally, although it was a while back, but a SARC inhibitor, which is expected to hit YES1 as well, was shown to reduce cisplatin hearing loss in mice as well. And this showing here, you have a change in hearing following cisplatin exposure, however, after day 5. But however, mice that were treated with a SARC inhibitor were protected from this adverse event. Now, of course, use of this is only going to be beneficial if OCT2 inhibition does not consequently sacrifice anti-tumor efficacy. Now, the great news about this has only been done so far with nilotinib, but cisplatin uptake seems to be unaffected by nilotinib exposure as well, or a combination with nilotinib. And this was shown using various lung cancer cell lines. And likewise, the sensitivity using cytotoxicity IC50 assessment shows that, in fact, while you don't have loss of IC50 or sensitivity, you actually, in some cases, increase tumor sensitivity to cisplatin in the combination with nilotinib, which therefore indicates this might be a fantastic combination where we can actually, in some cases, improve anti-tumor efficacy while potentially inhibiting toxicities. So, of course, future investigation is needed. A lot of what I showed you was preclinical models, but overall, I've shown you that tyrosine kinase inhibitors can impact transporter function, and this is largely through interference of tyrosine phosphorylation. Disruption of this phosphorylation event can alter drug disposition. And through this, you can have increased toxicity in some cases, depending on the combination you're giving. So, for example, OATP1B1 substrates such as statins, you may want to avoid tyrosine kinase inhibitors being given in combination with this. However, in contrast, tyrosine kinase inhibitor drug interactions can be beneficial in the case of such as OCT2 and platinum drugs, where we can protect accumulation or movement or transporters that move drugs into sensitive tissues through this combination. Obviously, understanding drug combinations can guide optimal treatment. So the more we understand, obviously we got to do more research to bring this to clinical practice, but we seem to be on track for something that better understanding means we can design a better strategy for patients to improve their outcome in the clinic. So obviously that kind of concludes what I was going to go over, but where I was going to say, obviously this is a largely a group effort, so I need to thank a lot of the members from my lab who generated a lot of the data that I've shown here today, as well as past members that have moved on from the lab. I also want to thank our collaborators, all shown here, from OSU, St. Jude Children's Research Hospital, Howard Hughes, Woodward Hoffman Research Institute, as well as various individuals from UB. And then finally, our funding sources from the University of Buffalo, School of Pharmacy and Pharmaceutical Sciences, the CTSI, ACP, NIH, and NSF. So with that, thank you for your time.